Hello, and welcome to the Astrolabe tutorial series. In this video, I will discuss how to explore the results of differential expression analysis for your experiment. This video will build on material presented in other tutorials. If you need a refresher, you can find links to tutorials about differential expression analysis and differential expression analysis setup below. For this demo, we are using a publicly available dataset from Glassberg et al., which used an ex vivo model to mimic the effects of thrombosis on a control and a patient group with sickle cell disease. There are several features of interest in this experiment, including control and patient samples, pre- and post-implementation of the model, and 3 time points at 10, 30, and 60 minutes when samples were taken. I have already set up this experiment for differential expression analysis, so let's examine the results by clicking on Analyses. Since I expect the effect of the ex vivo model will be most pronounced at the 60-minute time point, I'm going to start there. Let's just take a minute to review our equation from experiment setup. What we have asked Astrolabe to do is compare the difference between pre- and post-implementation of the ex vivo model between patients and controls. So when I look at this heat map, I can see markers across the x-axis and cell subsets across the y-axis, and each cell represents the fold change between patients and controls. The more saturated the color, the greater the fold change. To simplify this a bit, I'm going to come up here and select Compartment, which will roll my cell subsets into larger categories. In this view, I can see that myeloids seem to be of particular interest. To see some more detail, I'm going to select myeloids, and let's also select granulocytes. Let's also examine CD61, CD66B, pCreb, pERK, pMapCap, and pp38. If I scroll down, I can see more detail for each of these markers. And in each section, I will get a heat map with my sample names on the y-axis and my compartments across the x-axis. I can sort of tell by the shading here that the intensity of CD61 is higher in the control samples than the patient samples. But if I want to see that a little more clearly, I can select Feature Box Plots. Here I can see that the granulocytes don't show much variation at all between the patients and controls, but I can see pretty clearly for the myeloids that the intensity of the change in CD61 is higher in the controls than in the patients. If I would like a little more detail, I can select Sample Dot Plot, which really is the same as the box plot, except I can see which dot belongs to each sample. If I continue to scroll down, I can review other markers. CD66B actually doesn't seem that interesting, except for this potential outlier in patient 3. pCreb, pERK, pMapCap seem to have pretty modest variation between the patients and controls. However, the delta for pp38 is a bit more pronounced. If I go back to the assignment level and select myeloids in more detail, I can see that the variation is not really in NK cells, but in the monocytes. In fact, this observation was made in the study. Here is a chart from the Glassberg et al. Study that shows measurements for these markers in the control and patient groups. We can see those same modest variations, as well as the more pronounced variation in pp38. Something I found interesting about this analysis is that the study showed the platelet-rich thrombus formed by samples from the patients with sickle cell disease was larger. However, as we saw in Astrolabe, the variation of CD61 was less pronounced in those patients than the controls. That is something I would want to explore further. Next in this series, I will show you how to access your differential expression analysis reports. Remember, if you have any questions, you can always email us at support@astrolabediagnostics.com.